Hello, everyone, and welcome to today's live broadcast, Exploring the Genetic Landscape of Solid Tumors Using Whole Genome Copy Number Analysis, presented by Dr. Ravindra Kolhe and Dr. Joanna Przybyl. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Thermo Fisher Scientific. For more information on our sponsor, please click on the Thermo Fisher Scientific logo on the left of your screen. Now let's get started. I want to remind everyone that today's event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing the presentation, click on the Support tab found on the top right of the presentation window, or report your problem by using the Ask a Question box. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of your presentation window and follow the process to obtain your credits. I would now like to introduce you to our speakers, Dr. Joanna Przybyl, Senior Research Scientist at Stanford University. Also speaking, Dr. Ravindra Kolhe, Associate Professor, Pathology, Associate Director, Residency Program, and Medical Director, Cytogenics Laboratory, AUMC. For complete biographies on our speaker, please visit the Biography tab at the top of the screen. Dr. Przybyl, you may now begin your presentation.
Hello, everyone. Thank you for participating in this webinar. Today, I will tell you about the use of SNP arrays for studying genomic profiles of tumors with complex DNA copy number alterations, called leiomyosarcomas. Leiomyosarcoma is one of the most common types of soft tissue sarcomas, tumors arising from different types of connective tissues. It's a malignant type of tumor with smooth muscle differentiation. The most common locations of leiomyosarcoma include retroperitoneum, extremities, and the uterus. Leiomyosarcomas are associated with poor clinical outcome, and current therapeutic options provide unsatisfactory results. Also, the assessment of response to treatment in leiomyosarcoma patients is difficult since the conventional imaging may not provide informative results. Because of that, new tools for an improved evaluation of response to treatment is highly needed for patients with leiomyosarcoma. Leiomyosarcoma tumors are characterized by high genomic instability. On the picture on the left, you can see an example of a normal karyotype of a male with 2 copies of each autosomal chromosomes and 1 copy of each sex chromosomes, X and Y. On the right-hand side, you can see a karyotype of a representative case of leiomyosarcoma tumor from a female. And in this image, you can see additional copies of multiple chromosomes and losses of selected chromosomes. The most frequent DNA copy number losses in leiomyosarcoma affect multiple tumor suppressor genes like p53, RB1, PTEN, APRX, or CDKN2A. On the other hand, the most frequent copy number gains result in amplification of myoCD gene that encodes myocardin, a muscle-specific transcriptional coactivator. This graph summarizes the median number of somatic point mutations and copy number alterations across different types of cancers. And based on this graph, you can see that uterine leiomyosarcomas, as well as leiomyosarcomas arising in different types of soft tissues, are characterized by intermediate levels of both DNA copy number alterations and somatic point mutations. The most important challenges in the treatment of patients with leiomyosarcoma are related to the lack of therapeutic targets in these tumors and also inadequate methods for evaluation of response to treatment. In this presentation, I would like to tell you about our liquid biopsy studies in leiomyosarcomas based on profiling molecular alterations in tumor-derived DNA in circulation. Liquid biopsy approach may be potentially useful, especially for an improved and non-invasive assessment of response to treatment in patients with sarcomas. Potential applications of circulating tumor DNA include screening and early detection of cancer and improved diagnosis through genotyping of circulating tumor DNA. Potential applications include also detection of minimal residual disease after the surgery, evaluation of response to adjuvant therapy, and detection of genetic alterations that may be associated with resistance to certain types of treatments. And finally, circulating tumor DNA profiling may be also useful for a long-term follow-up in cancer patients to detect disease recurrence. Circulating tumor DNA is found in plasma or serum fraction of the blood. It's released by cells dying through apoptosis or necrosis, or it can be also actively secreted in exosomes. It has been shown that DNA in circulation may harbor the same molecular alterations as DNA in the tumor. These alterations include point mutations, copy number alterations, chromosomal rearrangements, and DNA methylation. The clinical utility of circulating tumor DNA monitoring has been shown in tumors that harbor highly recurrent mutations. As I have shown you, leiomyosarcoma represents a type of tumor with a wide spectrum and intermediate spectrum of point mutations and copy number alterations. Therefore, we proposed a new strategy for profiling of circulating tumor DNA in leiomyosarcoma patients, and that approach combines detection of point mutations by deep targeted sequencing using a custom capture panel, together with detection of copy number alterations through shallow whole genome sequencing. We assume that such combination approach may allow for an increased sensitivity of detection of circulating tumor DNA and may allow to track a higher number of markers in plasma. For detection of DNA copy number alterations, we profiled DNA extracted from tumor specimens using OncoScan FFPE assay, and we profiled DNA from plasma by shallow whole genome sequencing. From each patient, we profiled up to 7 different tumor specimens and several longitudinal plasma specimens. An essential step in this process is selection of appropriate FFPE tumor specimens. First, we evaluate the percentage of tumor cells in the specimen based on HNT staining. For this particular study, we selected mostly the samples with 90–95% of tumor cells. Next, we performed microdissection of the FFPE tumor specimens by taking several 2mm cores from the area with high content of tumor cells. After that, we extracted DNA from these 2mm cores using all-purpose DNA RNA FFPE kit, including RNase treatment. DNA concentration and yield was evaluated by qubit. Specifically, we used the high sensitivity double-stranded DNA assay. And later we submit at least 80 nanograms of DNA extracted this way for OncoScan FFPE assay to Thermo Fisher services. The first step in the analysis of the OncoScan data includes evaluation of a variety of quality control metrics. Selected important QC metrics include the median absolute pairwise difference, MAPD, which should be below 0.3, and also SNP quality control of normal diploid markers, which should be above 26. Diploid recentering may be performed manually if needed, which may be the case especially in the tumors with high genomic instability. I will show an example of diploid recentering in the following slides. In order to identify copy number alterations in our leiomyosarcoma study, we defined copy number gains and losses as the regions with log2 ratio above 0.25 and below -0.25 respectively. We required a minimum of 200 probes per segment to focus only on relatively large alterations. And with this criteria, we identified a median of 80 alterations per tumor sample. Diploid recentering may be required in tumors with high genomic instability that may have low count of diploid regions. In such cases, normal diploid regions of log2 ratio equals 0 may not be recognized properly by the software, and it may be necessary to manually assign the diploid region of the sample, or in other words, to recenter the sample. Correct diploid regions may be identified based on the B allele frequency profile. In this example of leiomyosarcoma tumor specimen, you can see multiple false positive calls. For example, gains of chromosomes 1, 3p, or 4p that are represented in the top panel with log2 ratios. We call these gains false positive because they have no corresponding allelic imbalance, which can be seen in the bottom panel called BAF, B-allele frequency. To manually recenter this sample, we may provide an offset that tells the algorithm how much the profile should be pushed up or down so that the normal diploid regions reside at log2 ratio of 0. In this case, we indicate that chromosome 1 is in the normal diploid state. We use the median log2 ratio of chromosome 1, which originally was 0.62, as the new log2 ratio 0. And in the bottom panel, you can see how the log2 ratio of each chromosome has been corrected after diploid centering using chromosome 1 as the new diploid reference. In this slide, you can see genomic profiles of the sample before and after this correction. The upper panel shows the original profile with false positive calls without corresponding allelic imbalances. And in the bottom panel, you can see the profile with correctly assigned normal diploid regions. Based on applying diploid recentering and the analysis criteria that I described in the previous slides, we identified the following abnormalities in our leiomyosarcoma tumor specimens. The most frequent alterations included loss of RB1 tumor suppressor gene and amplification of myocardin gene, which is in agreement with the previously described leiomyosarcoma profiles in the literature. Interestingly, in the study, we profiled multiple tumor specimens from the same patients or multiple regions of the same tumor in order to study inter and intra-tumor heterogeneity of copy number alterations in leiomyosarcoma. In patient LMS2, for example, you can see that we profiled 2 regions of primary uterine leiomyosarcoma, which is indicated as T1 and T3, and we also profiled a metastatic tumor from brain. And based on genomic profiles of the samples, you can see heterogeneity within the primary tumor, which is indicated by selected copy number gains or losses that are being present only in certain region of the primary tumor and not in the other. For example, in chromosomes 1 or 2. In patient LMS5, we profiled the metastatic retroperitoneal tumor and multiple concurrent metastatic tumors from the abdomen that were removed approximately 1 year after the first tumor specimen. And in this example, you can appreciate also the inter-tumor heterogeneity in leiomyosarcoma, for example, by looking at alterations in chromosomes 1, 3, or 18 across different tumor specimens. In patient LMS6, we profiled two different regions of a primary pelvic leiomyosarcoma and 2 separate metastatic lung nodules. And from these profiles, you can see a big difference between genomic aberrations found in 2 separate metastatic lung nodules that are indicated as T3 and T4. And in patient LMS7, you can also see inter-tumor heterogeneity based on the analysis of the primary tumor from the calf and metastatic tumor from the thigh. After characterizing genomic profiles of leiomyosarcoma tumor specimens, we aim to identify tumor-derived alterations in cell-free DNA in patient-matched plasma specimens. OncoScan FFP assay was used to define DNA copy number profiles of leiomyosarcoma tumors because SNP arrays are currently the gold standard for detection and quantification of DNA copy number alterations. Shallow whole genome sequencing was applied to detect DNA copy number alterations in plasma cell-free DNA, but this method may produce many false positive calls. Therefore, in order to identify only tumor-derived alterations in plasma DNA, we selected only the overlapping genomic regions altered both in plasma and tumor DNA in each patient. Shallow whole genome sequencing was performed on cell-free DNA extracted from 27 plasma specimens with the median coverage of 0.21x. This allowed to identify tumor-derived alterations in 44% of the samples in 5 of 7 patients included in this study. Using this approach, we detected up to 194 tumor-derived DNA copy number alterations per plasma sample. Copy number profiling in circulating tumor DNA was complemented by targeted deep sequencing by CAPSIC, cancer personalized profiling by deep sequencing. Using this method, we are able to track somatic point mutations in tumor suppressor genes, such as P53, RB1, and ATRX. We detected mutations in these genes in 68% of the analyzed samples from 6 out of 7 patients included in this study. This slide summarizes circulating tumor DNA profiles in selected patients with longitudinal plasma samples analyzed. The green line indicates circulating tumor DNA detected by CAPSIC, so deep targeted sequencing. The dark blue line represents circulating tumor DNA levels detected by shallow whole genome sequencing, so the DNA copy number alterations. And the light blue panels in the background indicate the duration of chemotherapy. In patient LMS-free, you can see that initially high levels of circulating tumor DNA become undetectable after the initiation of chemotherapy and radiation therapy. However, circulating tumor DNA becomes detectable again with the progression of the disease. A similar scenario was observed in patient LMS-5, where circulating tumor DNA levels dropped after the surgery, radiation therapy, and ablation, but then the levels increased throughout the progression of disease. In patient LMS7, you can see that circulating tumor DNA became undetectable for at least 1 year after complete resection of the tumor. In these profiles, you can see that the levels of circulating tumor DNA correspond with respect to treatment in those patients. Finally, I would like to show how circulating tumor DNA may reflect inter-tumor heterogeneity. A major advantage of circulating tumor DNA compared to tissue biopsies is that circulating tumor DNA analysis simultaneously integrates contributions from multiple tumor deposits, enabling a more comprehensive analysis of tumor heterogeneity. In the left panel, you can see that the 1Q gain was detected by SNP array only in a single tumor that was removed during the second surgery. In the right panel, you can see that the levels of DNA with 1Q gain in circulation were detected at 2 different time points among 5 longitudinal plasma samples. In these profiles, we can see that the 1Q gain was detected already in the first plasma sample that was collected 7 days before the first surgery. But this DNA alteration was not detected in the tumor sample removed at that time, because we analyzed only a single tumor, a single region of that tumor. In summary, our study of leiomyosarcoma shows that OncoScan FFPE assays are useful for detection of DNA copy number alterations in archival tumor specimens. I showed an example of deployed recentering that allows for correction of genomic profiles in tumors with high complexity, such as leiomyosarcoma. Our results show that copy number profiling using OncoScan FFPE assays may reveal intra and inter-tumor heterogeneity. And in this particular study, genomic profiles characterized by SNP array in tumor specimens served as the reference for detection of circulating tumor DNA in plasma. Thank you very much for listening, and I'm going to pass it over to Dr. Kolhe.
Thank you, Joanna. Good morning. My name is Ravindra Kolhe. I'm a pathologist at Medical College of Georgia, Augusta University. And today I'm going to talk about the utility of SNP arrays and targeted somatic mutations in evaluation of histologic mimics of myeloma. These are relevant disclosures about the talk. I'm employed by Medical College of Georgia, and I do have support from this commercial entity. Additional disclosures related to the talk, but most importantly, the opinions expressed in this presentation are my personal and does not represent the opinions of Thermo Fisher Scientific and its affiliates or any of my collaborations and employers. The product OncoScan FFP I'm going to talk about is intended for molecular biology application and not for diagnosis or prevention or treatment of disease. As I mentioned earlier, I'm a molecular pathologist and I practice cancer cytogenetics along with that in the capacity as a medical director and the CLIA lab director. But I also practice breast and surgical pathology, and that plays a critical role in using these new technologies in evaluation of surgical pathology cases. This is my outline. I'll go briefly over the assay and SNP array as a whole, but I think Joanna did a phenomenal job in introduction to OncoScan. I want to focus on why we ended up choosing melanoma as a poster child for its utility, and I'll add some other histologically challenging cases the OncoScan FFP for. So before I begin, we all need to acknowledge the great service karyotype has done for us in the past 30 plus years. But along with that, we also need to recognize the limitations karyotype has, especially with the low diagnostic yield, the longer turnaround time, especially the low resolution, and it's really difficult these days to find these trained people who could help us with karyotyping, and as it is a subjective method, it's prone for human error. The other couple of challenges we see, especially in the surgical pathology field for karyotyping solid tumors, is sometimes we get these blinded samples in the frozen room, which yield basically a normal karyotype. Along with it, there's overgrowth of non-neoplastic cells, again, giving an inconsistent results. On top of that, if it's a necrotic tissue or an abscess which leads to a culture failure or microbial growth in karyotype cultures. So I want to add a couple of points to Joanna's introduction that the OncoScan MIP probes, which we use for the SNP arrays in the lab, has a pretty phenomenal coverage, more focused on the oncology targets on the array, talking about 240 case SNPs. But the icing on the cake, particularly, especially if you're working in oncology, are the 74 SNP probes for the somatic mutation detection. And these probes are directed against 74 mutations for 9 critically important genes in oncology, like BRAF, KRAS, EGFR, which are important in either diagnosis, prognosis, or therapy selection for majority of the cancer cases. So lastly, the need for microarray in cancer research is critical because as compared to other technologies we have been using, we don't need the dividing cells, just the DNA from the target neoplastic cells. The assessment of the genome is at extremely high resolution in SNP arrays. The interpretation is objective and is based on biostatistical algorithms, so there is a very less human bias. One of the great advantages of using a SNP array is detection of copy-neutral stretch homozygosity, or LOH, what we call them. And on the research side, it's a huge advantage because all this data is digital, and you could upload to genome browsers and get annotated another annotated database and get really nice informative information on N of 1 cases. So coming back to the discussion topic of why we ended up choosing melanoma as a poster child for the SNP evaluation. Everyone knows that the melanoma is a critical problem, especially if you live in the South or in the Sun Belt. The H&E stained tissue still remains the main approach for evaluating melanocytic tumors, so again, we are not trying to replace H&E with SNP or any other molecular testing. The critical part is not the diagnosis of melanoma, but it is separation of these histologic mimics of melanoma, which are a very problematic practical issue in dermatopathology. And the reason for that is when melanoma escapes early detection, it is one of the most aggressive and highly lethal form of cancers. Although melanoma is a small part of the skin cancer, it is actually the major reason of skin cancer-related deaths in the United States. So when I say about histologic mimics of melanoma, I'm talking about histologic entities in dermatopathology like atypical spitzoid nevus or spindle cell melanomas, which mimic AFX or FH type lesions, nevoid melanomas, proliferative nodules, or large, especially in congenital nevi, clear cell sarcomas, regressed melanoma versus melanosis, melanoma transformation, especially in dysplastic or atypical nevus. So these are the kinds of lesions which are histologically very challenging in a small or limited biopsy. And because of this limitation, we are aware that either melanoma is underdiagnosed or overdiagnosed, and this misdiagnosis of melanocytic lesion is at the top of the list of malpractice cases in pathology, especially in dermatopathology. So let's take a step back and look at these the progression of these melanocytic lesions, right from a melanocyte or a dysplastic nevus. There are four main processes: proliferation, immortalization, invasion, and metastasis. And the good part is there are critical changes both at the copy number as well as the SNVs, which are gained, added during this process, which are very specific for that particular moment. And people have been exploiting these changes to use in different types of assays to answer these questions of melanoma versus histologic mimicry. And one of the first tests introduced was the FISH testing to evaluate these cases. To look at the first generation of the FISH assays, which were used as a surrogate for these genomic instability. Again, this is the data coming out from 10 to 15 years of research on array CGH based on melanocytic lesion all across the board. One of the challenges of these first generation FISH assays was a very high cutoff for these particular assays, which makes it very difficult to rule out melanoma, especially in critical cases. Then the second generation of the FISH came where the new probes were added to expand the diagnostic utility, but still the cutoff for these lesions was, these probes was very high. Then, and as I said earlier, the challenge was the sensitivity and specificity along with cutoff for these sets of probes. The added disadvantage was polyploidy and tetraploidy, which was a common source of false positives along with these FISH in these lesions. Apart from the limitations in the FISH, we felt like melanoma had multiple other reasons, but at least these two to look at. One was the TCGA database gave out 4 new genomic subtypes of melanomas: the melanoma which are driven with BRAF, the one which are driven with RAS, NF1, and triple wild type. So I think at this day and age, we need to have this granular information along with the histologic diagnosis of melanoma to enable our oncologists to identify cases in these different groups. Also, CAP does have a pretty in-depth melanoma biomarker reporting template, which I think is critical along with the diagnosis of melanoma. So to summarize, the reason we chose melanoma because the ambiguous nature of this histologic mimics was very problematic. There were the existing limitations of the FISH testing, the CAP biomarker template, which needed more information, and the TCG provided with the new molecular subtypes of melanoma. So to summarize, these are the different genomic aberrations in different melanocytic lesions, which are captured across the last 15 to 20 years of research. And the idea was whether we can put them together to help us use a SNP-based technology and help separate these histologic mimics. So, initial evaluation using OncoScan FFPE assay, what we did was we picked up 50 cases of melanoma which were worked out in a reference lab on fish testing, out of which 36 were histologically mimics of melanoma which were proven on fish, and the 14 were fish-proven melanoma. 95 of these cases were punch or shave biopsies, with a small tissue, which is the realistic samples which we received from our derm clinic. All the samples in our study passed initial QC. All the samples were run on the OncoScan FFPE assay, and the data was analyzed on the CHAS 3.0 analysis software provided by the manufacturer. So in reality, we were looking at these 50 shades of melanoma because I'm going to show you another slide which tells you that all these different examples of melanocytic lesions and their range of complexity associated with melanoma and how challenging they are in preventing us from making a consensus diagnosis of melanoma. So these are some examples of the cases in our study, and each and every one possesses a challenge in making a diagnosis of melanoma. For the lab workflow, most if not all the cases, we get a block from our histology or derm pad. We look at the H&E slide, a macro dissection is performed depending on the tumor cellularity and the circumference, and then the DNA is extracted. So we use around 5 micron sections from FFP block, 2 to 3 sections depending on the size of the tissue, depaptonization, DNA isolation, the nanodrop QC check, and then adjusting the DNA volume to get a 12 nanogram per microliter concentration for the assay. So again, to summarize, the kinds of samples or histologic diagnosis and their numbers which we used in this evaluation assay was anything which range from atypical dermal spindle cell melanocytic lesion to something as a compound pigmented atypical spinzoid tumor with congenital features and features of blue nevus. So these are literally complex histologic mimics of melanoma which we used in our evaluation assay. Just to give an example of how whole genome view would look on OncoScan FFPE assay. This is just an example of a melanoma case which shows the gain and loss in the critical genes which are usually abnormal in the melanoma. And as I earlier mentioned, the advantage of an OncoScan FFPE assay is the probe against 74 mutations, and one of them is the beta P600E, which you can identify in the keratograph of melanoma. So the result of the evaluation study was pretty phenomenal. We were able to get a near 100% analytical sensitivity and specificity. But for me, the most critical value was the negative predictive value. So in the assay, we are not trying to make the diagnosis of melanoma, but we're trying to make sure that we are not missing melanoma in these histologic mimics of melanoma. So in addition to the melanocytic assessment of these cases, we use OncoScan FFPE assay for assessment of low-grade adult brain tumors, atypical lipomatous tumors, atypical lipomatous tumors. I think Joanna did a phenomenal job in explaining the critical importance of SNP arrays. More recently, we have started to evaluate the bile duct brushing FNA samples and see how we can use OncoScan FFPE in evaluating these sample types. I'm just going to go over quickly a couple of additional surgical pathology cases we are evaluating and helping our surgical pathologists. So one of the great example, I think this is now a standard of care in majority of the academic labs, is the low-grade adult brain tumors. One of the biggest advantage of using an assay like OncoScan FFPE is a combination of both CNVs as well as mutations. The NEJM article, again based on the TCGA database, basically identified the limitation of histologic diagnosis, saying that the low-grade gliomas, if you have an IDH wild type of 1p19q codeletion, have a whole different clinical presentation and outcome irrespective of the grade type, and that has led us to identify this newer generation of testing modalities to help our neuropathologists and neuro-oncologists. So I'm just summarizing the NEJM article, which basically identified this limitation in our surgical pathology diagnosis. So just to give an example how we evaluated the OncoScan FFPE assays for low-grade adult brain tumors, we go through a barrage of SNP areas, something as IDH1 and 2, BRAF600E, EGFR, PDGFR, meta-amplification, 1p19q deletion, and so on, which has helped us tremendously to do a proper evaluation of our low-grade adult brain tumors. And this is just evidence-based, publicly available algorithm which talks about subclassifying, the molecular subclassification of adult brain tumors on SNP arrays, especially in OncoScan FFPE. This is again a whole genome view of adult brain tumors, which looks at different genes which are dysregulated on an adult brain case tumor, which again is very critical in analysis for based on the new WHO classification of adult brain tumors. This is again a whole karogram view which adds the IDH1, 1p19q analysis to the surgical pathology evaluation of this oligodendroglioma case. In addition to melanoma and adult brain tumors, we also look at atypical lipomatous tumors, especially differentiating between something as simple as lipoma versus liposarcoma on a limited core biopsies. We have again looked at a barrage of abnormalities which are critical in making that differentiation, and this is just an example which we presented at the CEP meeting last year, which looks at these small core biopsies which are very histologically challenging, especially in a big retroperitoneal region. But the whole genome SNP array definitely makes a substantial addition to the histologic evaluation of these small biopsies, especially differentiating between lipoma or liposarcoma. Something very trivial on the cytology, like specimens where you're trying to differentiate between reactive mesothelioma versus mesothelioma, where a loss of BAP1, which can be detected on immunohistochemistry, but also on an LOH of BAP1, especially on 3p211, makes a huge addition to the cytologic evaluation of these, again, cytologically challenging cases like meso versus reactive meso. So these are the additional cases or additional surgical pathology scenarios where an assay like OncoScan FFPE or a SNP array can make a substantial addition to the histology or cytologic evaluation for the surgical pathologists or cytoppathologists. We use them for atypical lipomatous tumors, renal cell carcinoma, especially for histologic classification, especially when we get these small core biopsies for post-cryosurgeries. We have ongoing research project to understand the C and V in Barrett's esophagus, especially in cytology or smaller specimen. GYN oncology with synchronous primaries with metastatic lesions. As I earlier mentioned, we're also looking at evaluating this ability in biliary and pancreatic duct brushings. So this is a lab, Dr. Chaube, Dr. Mondal, and Dr. DuPont are the different people in the lab which makes all these things possible. And these are some of the research associates in the lab and the lab techs working on this particular project. Thank you. Christy?
Thank you, Dr. Kolhe and Dr. Przybyl, for that informative presentation. We will now start the live Q&A portion of our webinar. If you have a question that you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. Okay, let's get started. Our first question. Now, Dr. Przybyl, Dr. Kolhe, I believe this could be for either of you. How many SNPs are covered by the OncoScan FFPE assay?
I can take this question, actually. So the OncoScan FFPE assay captures alleles of over 220,000 SNPs across human genome. And in addition, there are around 900 cancer genes that are covered with increased density of probes for better resolution.
Thank you. Now, Dr. Kolhe, I believe this one's for you. What is the DNA quality and quantity required by the OncoScan assay?
In our evaluation studies and currently in the lab, we try to get as much as possible, but our minimum cutoff is 80 nanograms of total DNA with the 12 nanogram per microliter as input volume. And it is measured by qubit, both double-stranded, and that something as that simple has been pretty successful in our assay.
Thank you, Dr. Kolhe. Dr. Przybyl, let's come over to you on this one. How were the DNA copy alterations identified in plasma cell-free DNA?
We applied the protocol that was initially developed for non-invasive prenatal testing by our colleagues from K11 in Belgium. And in brief, we performed single-end sequencing on Illumina machine, and we required at least 10 million reads per sample to be appropriately mapped. And we identified copy number alterations in plasma cell-free DNA using modified plasma-seq algorithm. And what is important for this analysis, we used almost 400 profiles of healthy donors to define what are the baseline profiles in healthy individuals. And based on this baseline, we defined genome-wide segmented disease scores in samples from leiomyosarcoma patients in relation to those normal profiles. So that's how the copy number alterations were called, based on the z-scores calculated from plasma cell-free DNA.
Thank you. And let's go ahead and have you answer this one as well. What type of specimens do you typically use?
The samples that we typically use specifically for OncoScan FFPE assay are sarcoma samples, because this is what our laboratory is focused on. So during my presentation, I explained exactly what is the process, how we select the samples. We pay a lot of attention to high content of tumor cells that is first evaluated by pathologists, and then I think that also the step of macro dissection of the sample is very important to avoid contamination from normal cells.
Thank you. And Dr. Kolhe, I'd like to ask you the same question. What type of specimens do you typically use?
So, Christy, in our evaluation study as well as currently in the lab, we use multiple different specimen types, mostly FFPE. In the FFPE, we use samples coming from punch biopsy, resection specimens, excisional biopsy. And as Joanna said, most, if not all, of these samples get H&E examination by a pathologist to assess the tumor cellularity and tumor enrichment, and then we do the macrodissection for DNA isolation. The samples are coming from a variety of tumor types like melanoma, adult brain tumors, leiomyomas, leiomyosarcomas, any specific histologic subtype where we have a challenging case, we tend to get these samples. More recently, we have also tried to evaluate the assay on cytology specimens, so these will be fresh FNA samples. And at the end of the day, what matters is a good quality and quantity of DNA. If we can achieve that, I think the assay performs well.
Thank you. And it looks like we have time for one more question. And Dr. Kolhe, let's end on this final question. What genomic region was analyzed in the targeted deep sequencing of plasma DNA?
I think I'm going to let Dr. Joanna answer that because I think her lab has done the work on the plasma DNA. Joanna, you want to take this?
Yes, of course. So for this study, we designed a custom capture panel that covers more than 180 kb, and this was specific panel designed based on the analysis of 77 leiomyosarcoma tumor specimens that were analyzed by TCGA consortium. We used exome sequencing data from these specimens paired with normal DNA from those patients, and that way we identified more than 300 exons that were the most frequently mutated in those patients, and that's how we came up with our custom panel for deep targeted sequencing of the most frequently mutated regions in leiomyosarcoma, and that's what we used to profile cell-free DNA from plasma.
Thank you again, Dr. Kolhe and Dr. Przybyl. Do you have any final comments for our audience?
I would like to just summarize by pointing out that OncoScan FFP assay is very useful for detection of copy number alterations in archival tumor specimens, and specifically it's also useful for characterizing tumor heterogeneity in tumors with high genomic complexity.
And then to follow up on Joanna's comments, I think, Christy, what OncoScan FFP assay has done in surgical pathology is helping our surgical pathologists to you do a pretty decent molecular analysis on histologically challenging cases like the one I mentioned in my talk. But on top of that, it is also helping us to provide a better diagnosis and prognosis of cases like adult brain tumors to match up with the new classifications, which are focused based on molecular subtypes.
Now, before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. I'd like to once again thank Dr. Ravindra Kolhe and Dr. Joanna Przybyl for their time today and for their important research. We'd also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Thank you for joining us, and we hope to see you again soon. Until next time, goodbye.